Great. Thank you so much. It's wonderful to be here. I think they put me first because my talk is a bit different than everything else on the rest of the agenda for the next 2 days. So we'll kind of knock mine out and then go on to a lot of the CAR T-cell therapy work. But as was mentioned here, what we're working on is a set of multi-cytokine inhibitors to immunomodulate patients so that it results in treatment for T-cell malignancies as our initial foray into the field. So the premise behind this was the idea of our CEO and founder, Dr. Nazli Azimi, while she was at the National Institutes of Health. Because one of the things that's known is that there's there's two ends of the spectrum when you're trying to knock out cytokines. There's this single cytokine approach, which is typically monoclonal antibodies, and on the other side you have the JAK inhibitors or PI3 kinase inhibitors, something like that, which is relatively non-specific, so it shuts off a large number of cytokines, which carries with it the penalty of safety issues. And so the idea was that there must be something in the middle to be able to manipulate, the disease-relevant cytokines for a given disease once that's understood without overdoing it, and so creating a more effective therapy, but also one that still retains the safety. So the first group of cytokines, the family of cytokines that we've been working on is the IL-2 family of cytokine, which is, I'm sure, all very familiar to all of you because of its role in the regulation of the normal immune system for T cells, B cells, and all the other cells that are constitutively made up in the immune system. So we're looking here, and so the issue here, and the beautiful part of this is that these 6 cytokines here share a common receptor subunit here, what's called the gamma C. Right, so you see within each of the 6 here, and they have their own heterodimer or heterotrimeric components that make up the ultimate receptor. And what we've done is we've developed a peptide therapeutic that actually is able to bind to the gamma C and selectively block only some of the cytokines while leaving the other cytokines unimpaired. And we've done this by designing a relatively small peptide, 24 amino acids, which is about 1/5 or 1/6 the size of the endogenous ligands of these cytokines. So it fits into the groove of the binding and results in inhibition of signaling for IL-2, IL-9, and IL-15. And you can actually do studies to look at signaling and downstream markers to show that there's absolutely no impact on IL-4, 7, or 21. So it's a very unique and selective approach to knock out these disease-driving cytokines, which fit into a number of the T-cell malignancies and some other autoimmune diseases that we're also investigating. We have pegylated it because if you give a peptide without pegylation, it'll be gone from the system in a matter of minutes. So once you put a long PEG molecule on this, it improves the pharmacokinetics to allow it to be dosed, once a week or once every other week. The beauty of this is that when you think about the cytokine signaling pathways, there's actually 3 intracellular signaling pathways that make up cytokine signaling. The JAK-STAT pathway is probably the most well-known and the most referenced because there are some approved therapeutics out there that target the JAK-STAT pathway. There's also a PI3 kinase and the ERK-MAP kinase as well, which is also signaling. So if you try to block the signaling of the cytokine using a JAK kinase inhibitor shown with the number 2, you're only shutting off one of the 3 components of signaling, so it creates a leaky, incomplete block of the cytokine signaling. So if you believe it's a pathologic cytokine and you want to block it, you really need to go upstream to the receptor, which is where BNZ1, which is what we call our compound BNZ1, works. And so by doing that, you're actually able to shut off, and this is some in vitro work looking at CD4 cells that are exposed to IL-2. You can see activation, phosphorylation of the STAT molecules, as well as the MAP kinase and the PI3 kinase. You do that in the presence of BNZ1. You see complete inhibition of all 3 signaling pathways. You do that same experiment now in the presence of ruxolitinib, which is a JAK1/2 inhibitor, and you can see it does a very nice job blocking the STAT activation, which you'd expect, but does nothing for the MAP kinase and the PI3 kinase. So if you really believe that it's important to shut this cytokine signaling off, you're only getting a partial effect from inhibiting it with the JAK inhibitors. So if you think about where we go with IL-2, 9, and 15 inhibitors, here's a table that shows a number of the T-cell malignancies that we've been thinking about that have cytokine involvement that's been documented by a number of different laboratories around the US and the world. And the ones that we're focusing on to begin with are these two at the top. Large granular lymphocyte leukemia, which has no FDA-approved therapies, which is shown to be IL-2 and IL-15 driven, and cutaneous T-cell lymphoma. And we're really focusing on the refractory CTCL patients. There are some approved therapies out there, but they don't really work. So there's some real opportunity to help patients by developing something that's more effective with a good safety profile. Once we develop or show proof of concept in these, we're looking at expanding into kind of an all-comers trial that would then evaluate the utility of Benzi-1 in these other T-cell malignancies that have similar or related cytokine-driven pathologies with them. So we've done some preclinical models looking at this. So this is a preclinical study looking at LGL-like IL-15 dependent tumors injected into mice, or wild-type mice who are then treated. So it's more of a prevention model. The tumor hasn't been established here. So we treat the mice with control saline, tofacitinib by osmotic mini pump, which blocks IL-15 signaling, as I showed, Benzi-1 at low dose and high dose. And what you see at the end of the study, as you go out 4-week study, that by the end of 4 weeks, the animals in the control and the tofacitinib treatment group have all died, while all the animals in the Benzi-1 treated group were alive. And even more surprising is that when we did immunohistopathology of these animals at the end, there was actually no trace of any tumor anywhere in the bodies of these animals. So it really shows a tremendous blockade of the IL-15 proliferation in this study. This next study is another preclinical model, but this is using ATLL cells, a much more aggressive tumor, HTLV1 driven. It's also IL-2 and IL-15 dependent. And so these are patient-derived cells that are injected into the animals. You allow it 2 weeks to develop, and here you can see at 2 weeks using fluorescence imaging. We now have some other treatment groups. We have the control, the Benzi-1 and daclizumab, which is an anti-IL-2 monoclonal against ruxolitinib, a JAK1/2 inhibitor. And what we were looking at initially was looking at IL-2 receptor alpha shedding as a marker of tumor growth. And you can see that it is going up in all 3 treatment groups except for the Benzi-1, which only starts to go up once you stop the treatment. And then when you look at survival, what you see is that the survival was better with Benzi-1 in comparison to either of the other 2 agents, which block incompletely and only one of the cytokines. So it shows that blocking both of the cytokines leads to a better outcome in this study. So we did our first studies in healthy volunteers. We went through this single ascending dose and multiple ascending dose studies, which are shown here. You start with a low dose. We started with 0.2 as our lowest, went up to 6.4 mg per kg. Single dose, we collected biomarker samples at 3 different time points that used for flow cytometry. You can see the PK graphs here is a very nice, it's an IV infusion. It takes about 30 seconds. It's a very quick, slow IV push, so it's not a long infusion that takes time. And you can see it goes up very nicely, and the clearance is very linear over time. So it's about a half-life of about 5 days, which is very supportive of a once a week or once every other week dosing. So based on this initial safety exposure, we went into a multiple dose study, went up with weekly dosing for the first 3 cohorts here, but also had every other week dosing shown with these last 2 here. And you can see that, again, the PK profiles are very well behaved, half-life about 5 days, so no change with multiple doses. The overall safety profile was really pristine and almost surprisingly so. Very few side effects and no relationship with dose. And what we're able to show in these healthy volunteers, luckily, is using flow cytometry. You can show that we were able to look at NK cells, which are IL-2 and IL-15 in the blue bars, Treg cells in the orange, and central memory CD8s in the gray. You can see a pretty nice exposure response effect with it, which was replicated in our multiple dose study. So whether it was single or multiple doses, you can knock these 3 cell populations down and absolutely have no effect on B cells, monocytes, overall T cells, or anything else. It's a very selective modulation. So what we're trying to do now is develop lower doses for autoimmune indications and higher doses for the cancer. And so this is our mechanism of action that we believe in the universe of T-cell malignancies. So you've got your malignant cells, the regulatory T cells, and the CD8s. We bring in Benzi-1. It blocks IL-2/15, get rid of the Tregs, and allow the immune system to participate in host attack, and then to also take care of the CD8s, which are part of the symptoms in these patients. So it's a very unique multi-pronged approach. This is the design of the study, dose escalation. We're on the 3rd cohort right now. It's been very well tolerated. We're starting to get some results out of it right now. I'll quickly, with my last seconds here, show you what happens in our first patient studied here. A very refractory patient. These are all their prior therapies, mycosis fungoides. And what you can see now is actually we identified that PD-1 expression can actually be knocked down in these patients because IL-2 and 15 are involved in upregulation of PD-1 expression. So we saw a pretty nice PD-1 expression that went down, and then we also saw that the CD8 T-cell activity measured by perforin expression was also suppressed and could be maintained out to day 28. And in this patient, we started to see some clearing of the lesions on the interior of the lesion right here, which is only after 1 month of treatment at our lowest dose. So we're pretty excited. We've got some more photos coming in from other patients that we're really starting to see some resolution. So we think we're kind of on the right track with respect to the patients with CTCL. So we got a very different mechanism of action. We've got a good safety and tolerability profile. We think there's a lot of opportunities to use our platform technology to develop other cytokine inhibitors focused on IL-2 alone or IL-15, IL-21, things like that. So we're really excited about where this is heading and hope to have the final results from this study by mid of next year. So thank you for your attention. And if you couldn't tell by my orange, I'm a Longhorn fan. So thanks so much.